Thank you very much for the introduction. Thank you for inviting me here and having me here to talk a bit about my research, which is so two spoilers. I'm not talking much about it's linked to what we heard before. I'm talking much about today about molecular simulations and therefore about clustering and dimensionality reduction, as we have heard before already. So it's less about deep learning. So you might argue that this talk is not very deep, and whether it's intelligent, we will see at the end. So when I prepared this talk, I was really wanting to talk about highly flexible simulations we are dealing with. But in order to compare them and why I actually tripped over this problem, I should give you a short introduction what my daily life with not so flexible systems is. So I would try to bring across a slight comparison of a system which works very well with very standard methods of analysis and dimensionality reduction, to then go on to explain a bit about a citizen science project actually we have, which has a lot of data and a lot of problems with analyzing these very flexible systems. So to give you a background, once again, thank you for the introduction. So I'm very much interested, since coming to Nottingham in particular, into industrial biotechnology. I joined a group of academics in the engineering department, actually, in Nottingham, who's very interested in using enzymes and bugs to produce chemicals, to digest plastics, so all the problems we have for sustainable production or sustainable degradation of products and waste products, and revalorizing waste products. And I'm basically a computational enzymologist in the group. So I'm very interested in biocatalysis. So every time we have to deal with an enzyme, so how does it really work? Because we need to understand how it works if we then want to go the next step, which is a rational design of these enzymes to make them better catalysts, to make them different catalysts, to make them work, for example, at high temperatures, to make them work in other than aqueous solutions if we want to do that. So within that, what I'm very interested is actually, can you see my mouse here? You can, I think. Is workflow development. So a lot of my work is related to molecular dynamics, but in enzyme catalysis and multi-scale modeling methods of using so. So what we need to do is we want to have a target. We have a target biocatalyst, which now should produce a different product or should work differently. So what my colleagues do in the lab, we have a lot of methods of to generate mutation libraries of these enzymes and then test them in the lab. One of the most well-known experimental techniques is their directed evolution. Highly efficient, highly costly, and still we haven't interpreted why, for example, in the end, a catalyst is working particularly better than the other one. With our computers, obviously, we have the chance to do this in silico. So I can also produce a mutation library, and with some knowledge behind that, I can rank my mutations for what they might what effect they might have. And this is where increasingly, so in this talk is a bit about also what I want to do and haven't done yet. So is that there is obviously some decision points which can be machine learning driven, which go into these method development, which go into these workflow developments. And if you see the acronyms down there, this is, for example, having really a look into what a specific mutation has an effect on my kinetics, my thermodynamics, on the whole complex picture of biocatalysis. To give you a view on that, where we are particularly interested in is my postdoc, Marco Hansavacci. He's really into radical enzymes, and I'm as well, by the way. But he is into a particular class, which is glycine radical enzymes. They are very interesting because they deal with peptide radicals, and every chemist knows that radicals are particularly nice in the lab because they are so reactive. So they are really hard to control. And we have a bunch of enzymes which very specifically can control them in a very, very nice manner. So the question is, from the biotechnology side of things, is can we actually make use of it? Either for us to make new products, or also these enzymes are very important in our gut microbiome. So from antibiotic resistance purposes, these could be also very interesting. These could be very interesting targets. So if you have a look into those, what we know is what drives biocatalysis, and we've worked a couple of years now on that. So we know, for example, how they control the intermediate radical stability is very important, and we have some work on that. We know for these radical enzymes in particular, and the enzyme activation of these biocatalysts is very important. And the other part of the equation is we know that the electrostatic environment, or better, the electric field inside the active site, has a strong effect on the kinetics of the reaction. So you see thermodynamics, kinetics, we work a bit, and you see from here I have 2 papers associated with that, but none with enzyme activation, because that's really a particular problem. But for example, for the to show you how this should go into the workflows, what we have recently done with Sam, what Sam has done during his PhD now is he looked into model systems, because the activating enzymes of these GREs have iron sulfur clusters in there. They are extremely redox active, extremely oxygen sensible, but we know that through the class they have a very different reactivity. So if we know what affects their activity, we can put this information into a design process. And he has done the first steps into that by what you see in this video, if it's playing, I think it is that he systematically looked via ab initio methods into what does a specific orientation of an electric field has an effect on its reactivity, on the redox reactivity of these clusters. So now we have a big data set we know what orientations of electric fields have an influence, and now we could put this back into a workflow where we look into mutations in the enzymes and see what field we have and link it then directly to this information. So that was just the introduction into that. Now we go into the activation of the glycine enzymes, because that brings us a bit more now into the dimensionality reduction and a bit old school machine learning methods, let's say it that way. The problem with these GRE enzymes is the activation is by another enzyme, which is the GRE activating enzymes, which is called, so you see it here in blue, and the big one, the greenish one, is the GRE itself. But if you think about, so again, chemists and biochemists know, if we think about a lock and key principle, we would say, okay, we dock these two together and then we have a look how this works. But it's not that easy for these enzymes. We know that the lock and key principle doesn't work here. There are extremely large conformational changes necessary that these enzymes bind at all. But we need this data if we want to input enzyme activation in our workflows. Again, we need this data. We need to know how these how this activation process works. So there is another thing we want to do and haven't done so. So what we need to do is we need to find a lot of conformations of these enzymes to try to dock them together to see whether they are stable together. And this is where we want to design another workflow where we actually go. We have a lot of a high population of possible simulations which lead to possible activation complexes, but I don't know which one it is. So I take a big population of structures, and then I start simulations, and then I gradually want to throw out simulations and replace those simulations by simulations which are more likely to finally lead me to what I want to see, okay? Which is the activation complex. And this is where neural networks can play a role again, because we can train them to actually give us a fitness score in the end of these simulations. But what you can already see, again, in order to train this, we need to have a supervised training method. And for a supervised training method, we need a data set. So in the first place, we need to generate all these structures and have a ranking mechanism for them already in place. And this is where lots of my work goes to. So we go to molecular dynamic simulations, a bit of enhanced sampling simulations, because we want to see, and what you see here is the GRE. We want to see whether we find a conformation which might actually bind. And if you see this wriggling about, you shouldn't see much. You see that I already highlighted a region in green for a good reason, because that is the region which is binding to the activating enzyme. And in purple down there, this blob is actually the residue which needs to be activated. And you see it's highly buried completely buried in there. And this is microseconds of molecular simulations, so it doesn't happen much at first sight. This is where the first dimensionally reduction comes in. So from this wriggling, we don't see it much, but actually if we apply principal component analysis, and yes, this is not yet, let's say in the 80s, you could have called it machine learning. Now, let's name it the poor man's machine learning approach. It's really a good dimensional reduction method, which, and this why I mentioned this, I come to this later, which relies on linearity in your system and linear transitions. And I just want to show that for this system, because here it works. And for many of my enzymes, it works. So the principal components see me the largest movements, linear movements in my data set. And if I look at this more closely, and I'll show you a video, these minima you see in here, so the most highly populated region in this space of PC1 to PC2, are actually really showing me the closed versus a more open structure. And what I then can nicely do, because we chemists want to see things rather than just having our data, is I can actually visualize this principle along my trajectory of data only along this principal component. And then I see a movement which is rather important, and you see this hinge is going out, and all this region might become approachable for the activating enzyme. And this is now what we then take into docking, and this is how we now can slowly build our data set of initial structure for our workflow we want to develop. But why I showed you this is, indeed, it is about, so we have a dimensional reduction method, PCA, which is often used for molecular dynamics simulations, and it works most of the time very nicely. And now I want to go to the example about our highly dynamic systems, and from this boiler, you might already see it might not work that well here. So why I'm talking about this is, what I'm talking about here is mycolic acids. So mycolic acids are lipids, relatively complex lipids, which are relatively big lipids, which are incorporated in the cell wall of Mycobacterium tuberculosis. And they have a specific role in there. They are part of the integrity of the cell wall. They are part of the defense of the cell wall, for example, against antibiotics. So they have a key role in antibiotic resistance. But it's not one molecule which has this role, it's a composition of different molecules. So these lipids are very diverse, and we know in nature a couple of 20 to 30 already, which we already discovered. But if you look at this chemical formula and the chemical space, there are hundreds of possible of these lipids. And key information here is how these lipids interact, how the lipids fold is important, how they build up the cell wall, or this compartment of the cell wall, better to say. So we simply wanted to understand how they fold, but we have quite a few of them, and we need to do quite some simulations. So this is where we paired up, this is wrong. So what we already know from monolayer experiments, is a couple of conformations, how they might fold, which is called the WUS conformations, because it either W or U or a Z type shape, so it's after some letters. And we know that depending on the compositions of different mycolic acids, how they interact with each other, different types of cell wall resistance can be engineered actually. So as said, we have a couple of these simulations. So we paired up and you have heard yesterday in the evening already something about citizen science. So we paired up here with the World Community Grid, which is using the BOINC engine, which goes back, I think, to the SETI, which was the first one when I was still in school, I think, a citizen computing. So where you get a package of data and you analyze. And it was radio telescope data to search for extraterrestrial life. And now the new World Community Grid, which was until last year hosted by IBM and is now hosted by Krembil, They are after humanitarian relevant disease-related citizen science. So they have a couple of projects. Ours is the tuberculosis project, but they are Zika projects, HIV projects, and not surprisingly also a COVID project now. So what we did here is really we constructed over 144 different mycolic acids, and we sent out over 80,000 simulations with different starting conditions because we really wanted to know whether these have an influence. We now have milliseconds of data to analyze. We could, before we reduce the data already, this system produces 500 gigabytes of data a day. But we can throw out a lot of information already at that stage. So at our end, it's only a couple of terabytes a year, which comes back. But as said, we have milliseconds of data all produced by a citizen science project. So the question is now, we just have a bit to analyze. And we saw XKCD comics before. They always work, particularly for data science, I think. Not a single talk, you can't use them. So yeah, we just have a bit of data, so let's analyze it, and you end up with the problem you actually create more data, and you can't even interpret it properly. So what I will show you in the next minutes here is why the problem for us is this. When I first started this project, or joined this project, I thought, these are lipids. Particularly in water, they fold into a structure. That's what they should do. But apparently, most of them don't. They are extremely flexible. They have very different conformations. They undergo an almost chaotic way of folding and unfolding and refolding during our simulations, which is also slightly different depending on the starting conditions. So we really need to analyze this data and need to find ways before we also have no chance of going through 80,000 simulations by hand. So how do we analyze these? But we already saw trends, obviously, depending on the chain lengths of these different and the chemical entities which are involved. So how do we go for it? And this is just a wrap up in 5 minutes. How long do I have, by the way?
You've got plenty. 10 minutes.
10 minutes, yeah. Fine. I wrap up in 5 minutes what we try to do, okay? And just a few take-home messages why it is important to go back and think about what was the reason a particular method failed. So I will briefly go through a supervised clustering method, which has nothing to do with machine learning. Go back to principal component analysis, show that initially it will show, oh, that looks good, but has some caveats. And then go into what we now just started, is using self-organizing maps to go through this data. So I showed you these conformations before, and you already see the problem with the standard clustering method. So what you can do, obviously, is you define branching points and you define distance thresholds and just go systematically through the data and cluster along that. And then you get what you put in. So you get the bias you put in, you get out, obviously, because you have predefined that only these structures which were known from experiments are in your data set. You only get these structures out, and that there's something wrong with it, with your analysis, you can see here in the last column, is actually that you only capture 10% of your simulation data at all. Okay? The rest seems to be not defined, which is not the case. So yes, it's simple. It's a simple principle. You can use it. But you have a low percentage coverage of conformations. It fails by definition because you put it in. And it fails to account for variability beyond this system, and it has no transferability at all. So remember that my data set, our data set, is complex in that sense that the molecules are not all the same size. So we have different chain lengths in between, which have a significant effect, different chemical functionalities as well. So now let's go to PCA, which is why I introduced it before. So when you use principal component analysis, the first step the analysis is doing for a Cartesian molecular data set, it transfers the whole system into internal coordinates. So we had a Cartesian space of the molecule wriggling around. We transfer it into a distance matrix of interatomic distances, which is easier to handle. and already removes freedoms of rotation and translation, which we don't want to cluster, obviously. And then by selecting which principal components we have, so the highest dimension of flexibility, which works very well for the key movement in the enzyme I showed you before, we can also apply it for here, and it looks very nicely in the beginning. The only next step we then have to do after we defined the principal components, we need to cluster along them, and this is what you see with these colored areas. And what you see in this picture is how we can do that easily. We just apply on the axis of the principal components, we bin them as histograms, and the density of the histograms, we apply a threshold again, and then we can define this cluster as here, the red sphere here. Or if we loosen the threshold, put it a bit up, then it's here. Okay, but we have clustering, the balance seems okay. The question is, do we capture the conformations we want to? And I come back to that in a second. By the histograms, obviously, we have a definition of energy in there already. The population gives me, by using a Boltzmann average, by definition gives me also the energy state of this. So I can visualize this nicely and can claim my molecule here is sitting in water and in hexane. It has different conformations in there, and the one is more stable than the other. Okay. The key problem with this is, if I show this, it would be shown to me, I would say, yes, this is a free energy difference. I already know which is the most stable. I have all the information I have. The thing is, we applied this dimensionality rejection, which is only looking for the principal components. So by definition of my principal components, of my largest movement, I have a free energy space on my largest movement, not on the whole space. So the question is, did I lose important information, or did I lose only noise? Because I only want to get rid of the noise. And this, again, it works good for the protein, because I'm only interested in the slow movement which is happening here, and the wriggling about is not so much. But does it also account for here? And the only way we can then elaborate this is by using other methods. But it definitely already works better. So we already see that the principal components, if you look at the picture below here, gives us new structures, which were the knotted structures we didn't have in any analysis before. So yes, it already reveals some hidden structures, and I can assign the percentage of folding for those. So the good thing is it finds new conformations. Problem is it needs individual inspection. And the other thing is it's still not transferable. So from molecule to molecule, my principal component space is still determined by the number of atoms, and therefore by the number of interatomic distances. So the transferability or comparability of different molecules is not that easy. So that's why we went then into using self-organizing maps for these types of problem, which is now a kind of, this is my 5 minutes, brilliant, which is a simple neural network. It is nice, and we had yesterday, or today as well, again, the discussion about interpretability. It has no hidden layers. It has a single layer of neurons. And I just mentioned here the 4 key hyperparameters, which is the number of neurons, the shape, which is normally only the decision whether these neurons are hexagonal or quadratic, which is how they interact with their environment. And a sigma value, which is the spread of the neighborhood function, and the learning rate. So I only show you the example for if we alter through different systems by changing the learning rate, which you can see here from left to the right. We have an enzymatic system. We have one mycolic acid on the bottom, and in the middle, we have a very interesting system, very flexible system. I didn't have the time to speak about today, which is a krypton system. Why I'm showing you that is what it shows us is that obviously by more training we get finer structures. We see a comparability. We see already by these pictures a discrimination between highly flexible and less flexible systems. And with that, we now have the opportunity, and we haven't done that yet, that we find the sweet spot of optimizing these parameters to then be applicable to all our systems. And this is why we've chosen this, because of the transferability. You can then get out the heat maps of your structures, and I don't go into detail here, but what we see is for those systems which are less flexible, which is the right-hand side, the KC29, which has a very, very much more folded structure, we see the same thing as with the PCA analysis. So there, it might have worked. For the more flexible, we again see a particular extended open conformations we have not seen in the PCA analysis. And as these are very interpretable, one single layer methods, PCA and SOM both are, we now can go back and say, and analyze why we haven't seen them in the PCA. So where, in which principal component, somewhere it might be, we have lost this data. So in summary, for these systems we have here, SOMs work quite nicely, and we need to go after it. We see more of the hidden structures. We have a systematic approach now, so we can automate the approach of optimizing our hyperparameters and then apply them to the whole data set without the need for too much visual inspection before we do this. And it's transferable because our net, the neural net behind that, is actually directly applicable on all the systems, and we are not determined by the size of the structure anymore. And it's not biased by large linear transitions in the data set, and I can go into detail, but not in this talk. A closing remark, because I won't have time to go through that, is another thing is we were looking at, and I can discuss this later in the break, is now what can we, before we analyze our data, our simulation data, can we actually predict what methods might be working better and which worse? And there are two things we can apply here, and one is actually chaos theory, where we can have something like, which is called the Lyapunov exponents, which gives us a tendency of chaos in our simulation, because even deterministic simulations can have elements of chaos in there. And yes, we can. I go into detail, but this is something we want to go through our whole data set again, whether this gives us reasonable data of also whether our folding is actually really deterministic or driven by numerical differences in our starting positions. That's why we have so many simulations. We also want to go after this. And with that, I want to thank everybody involved, particularly Connor. He's doing now the PhD and is going into the machine learning methods. Athena and Wilma started the whole project on mycolic acids. Marco is my enzyme postdoc, let's say it that way. Sam, a classical computational chemist, co-supervised with Jonathan Hurst in chemistry, who, as you have seen, did the work on the electric field catalysis. Anna Croft, my ex-mentor and still collaborator, and you for your attention.